Okay, good morning class. For today's lesson, we're going to be taking a look at how DNA replicates. And as part of our lesson, we're going to be constructing our own DNA models or DNA replication models using colored paper clips. So to get started, let's answer a few warm-up questions to review our previous lesson. So who can tell me what DNA is made up of? What chemical molecule is DNA made up of? Okay, that's right. It's made up of nucleotides. All right, and who can give me a list of the three parts that make up the base unit of DNA? Okay, correct. It's made up of a sugar, a phosphate, and a nitrogenous base. Okay, and then finally, who can give me the name of one person involved in determining the structure of DNA? Right, James Watson was one of the people involved in that process. Okay, so by the end of this lesson, each one of you should be able to explain how the process of DNA replication is carried out. All right, so let's take a look at this picture and kind of see how this whole process works. Okay, so as you can see from this cartoon figure here, we have a couple of enzymes that are involved in the process. Probably the main enzyme is this yellow one here, and this is DNA polymerase. This is the enzyme responsible for adding free nucleotides onto the separated strands. And then we have DNA helicase, which is involved in unwinding the strands and making them single-stranded. Okay, so the DNA molecule separates into two strands. Two new complementary strands are produced following rules of base pairing. Each strand of the double helix serves as a template for the new strand. Okay, and just to go a little further, replication is carried out by a series of enzymes that unzip the molecule. Two strands separate, forming two replication forks. DNA polymerase then joins individual nucleotides to produce a DNA molecule. All right, and then just to further explain how this process is carried out, let's watch this short video.
Is defined as the process by which an organism's original DNA is used as a template for the production of a new complementary DNA strand. An enzyme called helicase unwinds the original DNA's double helix, creating a replication fork. Next, an enzyme named DNA polymerase III works down the leading strand and up the lagging strand of the replication fork, synthesizing two new strands of DNA by taking free nucleotides and pairing them with the complementary bases on the original DNA template. The process of DNA replication is described as semi-conservative because the two copies of DNA produced each contain one strand of the original DNA and one entirely new DNA strand.
Was helpful. Now onto our activity. We're going to be making our own paperclip models demonstrating replication. So to start this activity, we're going to count out a series of colored paper clips. A's are going to represent adenine, yellow is going to represent thymine, red for cytosine, and green for guanine. Okay, so to get started with the construction, we're going to assemble a short sequence of human growth hormone gene. So take your paper clips and arrange them in the sequence outlined in this diagram below. Okay, once you've done that, go ahead and start unwinding your strands and forming your replication forks. Notice the rules of complementarity. A's pairs with T's and C's pairs with G's. Okay, now that you have your replication forks, demonstrate nucleotides being brought in to create 2 new strands. Complete the 2 new strands with your remaining paper clips. Okay, at the conclusion of our activity, you should be able to think about this question and complete it as homework. Compare and contrast. Take a look at your strands, compare and contrast how the new double-stranded models compare with your original model. Are their nucleotide sequences identical? Again, take this home, think about it, and bring it back with you for class tomorrow. Thank you.